Welcome friends. Today I would like to explain about plumage colors and patterns in chicken. What is plumage? Plumage is entire feathery covering of the bird. So plumage is a complex and important component of avian phenotype, and it is displayed by the arrangement of feathers with various pigmentation pattern. Specific colored feathers present on body of bird which provides overall distinct visual appearance of the fowl is called a plumage color. And definite arrangement of color on individual feather on the specific body region is called a plumage pattern. Feathers can get color by pigments like melanin, carotenoids and porphyrins called as chemical coloration. When these pigments get reflected, diffracted, dispersed or absorbs rays of light called structural coloration. So blue and green colors are structural in nature. When the feathers get colors due to both structure of the feather and pigments like yellow green and blue green, which are due to the combinations of chemical and structural colorations. The pigment melanin determines the plumage colors and patterns in feathers. This melanin pigment is divided into eumelanin and pheomelanin. Eumelanin is pigment of black, blue and grey color feathers, and pheomelanin is pigment of red, brown, salmon, buff color feathers. Distribution of these pigments is complicated by difference in feather form and structure associated with age, sex as well as structural variations between and within feather tract. So distribution of eumelanin in plumage causes primary or secondary color patterns. Now what is primary and secondary color patterns? Primary color patterns are due to the zonal or regional location of black pigment, that is eumelanin, and they may include several or few feather tracts, for example Columbian pattern or light feathers in pigeon pattern. Now what is secondary color patterns? So these secondary color patterns are produced by the effect of distribution of eumelanin within individual feathers, for example barring, lacing, spangling, etc. The another important thing to remember that the distribution of eumelanin and pheomelanin, which are subtypes of melanin pigment, appears to be controlled by a genetically determined competitive advantage of one type of pigment over the other. Therefore, plumage colors and patterns are polygenic in nature and controlled by multiple alleles at E locus and interactions with alleles at another loci. The gene interactions like dominance, incomplete dominance, and epistasis produces different color patterns in chicken feather. The primary plumage color patterns are extended black, birchen, dominant wheaten, black bistre red, that is called as wild type, then partridge and brown color. These primary color patterns are controlled by E locus that enhances the distribution of eumelanin in feather and depends on presence of allele at E locus. However, eumelanin restrictors, enhancers and diluter genes and pheomelanin intensifiers and diluter genes interacts with allele at E locus and also interacts with each other to cause zonal distribution of eumelanin as well as within individual feathers, which results in secondary feather color patterns. These secondary feather color patterns are penciling, barring, lacing, spangling, mottling, speckling, stippling, etc. In short, plumage colors are primarily decided by the eumelanin type of melanin pigment and depends on alleles at E locus and its interactions with allele at another locus, which forms primary and secondary color patterns in chicken.